So this here is a tiny water snail under the microscope, and as you can see, it starts to light up every couple of seconds. Why is it doing that? Well, I tell you, that is a little trick here. It's not doing it on its own, but if you want to find out why it's doing that or how it's doing that, then well, you'd have to watch until the very end of this video. So hi, hello and welcome again, Microbehunter here. I always keep my jars with water samples here on my window. Here I've got another jar, and this one contains not only a whole bunch of water fleas like ostracods and copepods and so on, but also some snails. And today I would like to put one of those snails under the microscope. Let's see if we're able to find something interesting. You can see the snails here on the inside of the jar. Yeah, there is, of course, a biofilm. There's some algae, diatoms and so on growing on the inside glass surface. And the snails, basically, they eat away this biofilm. They have plenty of food, therefore, in this jar, and they keep on reproducing. And yeah, they have various sizes, smaller ones and larger ones. New ones are born all the time, of course. Some of them die as well, and then the shells start to accumulate on the bottom of the jar. I think they might make an interesting specimen, and I have to basically now carefully try to catch one of them because when they come off the glass surface, then they sometimes fall down, obviously, so I have to somehow find them and catch them in such a way that they do not fall off, and some of them are kind of small, so they might be a little bit difficult to pick off. I'll be using this disposable plastic pipette to catch them. I cut off the tip to make the diameter a little bit larger so that the snails are going to fit in here. And there's of course the danger that when I try to extract the snails, extract is probably not the right word, but maybe when I try to remove them from the glass surface, there's of course the danger that they also basically fall through the opening of the pipette and then they're lost again. So what I have to do is I have to be very quick and I have to almost scoop them out at the same time as I suck up the water. You're gonna see what I mean. Yeah, I want to pick a smaller one here, or maybe the one up here, I don't know yet. Yeah, I caught it and I lost it again. Yeah, so you see a little bit the problem that I'm having. Let's try the next one. Yep, and here it is. Okay, yeah, it was fast enough, and now of course there's the danger, not the danger, but really the problem that it will start to stick to the inside surface, and then it's not gonna come off. Yeah, but if I keep on rotating it, then maybe I prevent it from actually attaching to the pipette. So let's quickly put it on a glass slide. Yeah, I have to first of all, of course, I have to remove the moss with the tardigrades, my slime molds and some other old slides which I need to clean. So, and let's quickly try to put the snail somewhere on here, and I lost it. Oh, where's the snail? No, I didn't lose it. It's up here. Here it is. Okay. Yeah, so how am I gonna do that? I need probably a Petri dish. Yeah, okay, so the snail is here, and I somehow need to get it out and then somehow on the slide. Maybe it's gonna be easier first to kind of flush it out here on the Petri dish. Nah, see, doesn't wanna come out. Here it is. Yep, a little bit of water quickly on the slide, and rest of the water goes back into the Petri dish. I think I'm just going to have a look now and see how it looks like. Yeah, so this here is not my stereo microscope, it's just my regular microscope using the 10 times objective, and it's huge, right? Yeah, let's see if we're able to move it here. So that is basically you can see the two antennae. It's a little bit blurry, of course, because the snail is fairly thick. Yep, and yeah, here we go. Yeah, the two eyes are visible as well. Here they are. Looks kind of now it attached itself to the surface, and now it's moving along, crawling along. Okay, yeah, here we go. Yeah, Okay, so that's basically the snail. Maybe I just want to go down with the magnification first. Okay, so here we go. I need to flip away the condenser. It's pulling along some I don't know some dirt and some other debris, and it's pretty dark actually. So let's turn up the light. But you see the light has a problem actually going through the shell of the snail, and so yeah. But in any case, you get the idea. I'm now going to put it under my stereo microscope. Yeah, and this is how it looks like now under the stereo microscope. You see it's a tiny snail. Maybe we can go up with the magnification a bit, but the higher we magnify, the lower the depth of field. So this means I have to refocus, and you see it's kind of difficult to get all parts in focus at the same time, and it's always moving out of the field of view as well. So maybe we're just going to reduce the magnification again. One of the problems with these snails is that their shell is kind of dark. Yeah, we're not able to look quite easily into them. At least the two eyes and the antenna are nicely visible. I'm going to now try to do the following. I'm going to flip the slide upside down, and I hope that I'm able to see the mouth of the snail. Okay, so I somehow have to flip it upside down, and maybe I'm gonna just support the slide with two additional other slides left and right, simply so that there is a little bit of a distance and a spacer. So I'm preparing the slides now. Okay. So the snail is now upside down, and yeah, maybe can go up again with the magnification a little bit, and yeah, this is how it looks like. I don't know. I don't see the so-called the radula. The radula, that is the mouth of the snail. A little bit maybe. And the mouth can be seen. Let me use the arrow. Can be seen over here somewhere, right? And with a radula, with the mouth, it's able to scrape off the surface, and it's harvesting this way. Yeah, the algae that are growing on the surface. So maybe I'm going to just change the lighting around, and I'm going to give it some bottom illumination. Yeah, this is now light shining through from the bottom, but it does not really reveal a lot of more details. Yeah, so yeah, not very good, unfortunately. The snail is simply a little bit too large, and maybe also a little bit too dark to see any details. Now, I'm not a snail expert, but according to research, many of these small snails belong to the genus Physa, and essentially what they have is they should have beneath their shell some kind of an air bubble that they use for breathing, because they are air-breathing snails. Yeah, I don't know so much about that. In any case, yeah, these snails are extremely common in the jar that I keep, and they keep on happily reproducing, and they are also the ones that are eating away the algae from the inside of the jar, and therefore they are kind of keeping the jar, the mini aquarium, also clean. Yeah, but this one here is now moving along, and yeah, maybe it's a little too small still. But you see, that's a little bit of a pity that I'm not able to see any more details here, because the snail itself is simply too dark. Yeah. And I remember a couple of months ago, I also put a snail under the microscope, and I could actually see beneath the shell that there was actually some air, but I'm not able to see that right now. Yeah. This particular individual is a little bit more transparent, and here we are able to see the air bubbles that are caught beneath its shell, and that's basically important for keeping the snail afloat, and also is an important oxygen supply underwater. So the snail that we have here is a so-called bladder snail. That is its common name. They're quite small freshwater snails, and they're quite common. And they are a little bit unusual from most other snails because their shell spirals into the opposite direction. They have been found in ponds, ditches, lakes and rivers, and of course also in aquariums. They are quite tolerant to polluted water, and this makes them quite common in many environments. Now, some species are indeed very common in aquariums, and they are considered even to be pests there. They spread by laying eggs on plants, and then these plants get reintroduced into aquarium tanks when you buy those plants. Some species reproduce quite quickly, and essentially they can even become a nuisance in an aquarium. However, it does not generally cause any harm to the aquarium. These snails, they feed on algae, detritus, that's decomposing material, and also the biofilm that grows on rocks and also on the aquarium window. And by constantly grazing, they help to keep the water clean and they prevent algae from growing too much. In aquariums, they can also help by cleaning the surfaces and breaking down organic waste and even, for example, dead fish. As anyone with a home aquarium knows, those bladder snails, they can reproduce quite quickly. Their ability to live in many conditions makes them also one of the most widespread freshwater snails. Now, I put the snail on a microscope slide and I removed most of the water. And then I could see that the snail retracted into its shell for protection. And then if you want to make it come out again, then you have to add again a little bit of water. And then I could see that it immediately emerged and it started moving around again. Scientists have identified about 80 species in this family. They are especially common in North and Central America, particularly in the Pacific drainage areas. These are the places where the rivers flow into the oceans. They were already described back in the 19th century, but their classification is still a little bit unclear because researchers have struggled to organize them properly. Some have tried to divide them into subfamilies and tribes and so on, but apparently there is no final agreement. Now, if I had a chance, of course, then I would conduct a systematic DNA study to better understand their biodiversity. Now, I would not solely rely on their appearance under the microscope for classification. Maybe such DNA studies have already been done. I'm not sure, but if you know anything, then of course, I think we'd all be very happy for some information in the comments below. Yeah, and what I'm going to do now is I'm just going to put it back again into the jar. So, and here we go. Okay. Yeah. Yep. Now it's gone again. It's back with its other friends here. I'm just gonna leave it at that. Hope that you liked the video. Yeah. I just like to look at things. That I find in my everyday environment to put under the microscope, and I hope I was able to share the passion also with you. Well, wish you all the best. Happy microbe hunting as always, and see you in the next video. Bye bye.